Great. So I'd like to welcome everybody back for the next session of this meeting, which is focused around inflammatory bowel disease. And our first speaker, it's with great pleasure that I'd like to introduce Carl Anderson from the Sanger Centre in Cambridge in UK, who's going to be talking about interpreting genome-wide association studies of IBD through the lens of single-cell technologies. Over to you, Carl.
Thank you very much, Alison. Just share my screen and put it into presentation mode. So yeah, I'd like to start by thanking the organisers for inviting me to speak today about some work that's ongoing in my lab aimed at using single-cell RNA sequencing to further the biological insights that we can make from genome-wide association studies of inflammatory bowel disease. I just want to start by actually acknowledging the folks in the group who are doing all of the work. So Rebecca McIntyre and Menno Guraba, who are working in the wet lab, developing the protocols for the single cell dissociation and processing all the samples. Monica Kuzak and Leland Taylor, two fantastic postdocs in the team who are handling all of the single cell RNA-seq data, undertaking the QC and doing lots of the analysis, including the differential gene expression analysis. Tobia Legby, who is doing all of the heritability enrichment analyses, which I'll go on to present later in the talk. And of course, the clinical lead, Tim Rain, who is leading the team, ascertaining all of the samples from the IBD patients and the healthy controls. And there's a large team of people who are involved, some of whom are listed here. Of course, we have to acknowledge the funders, and actually, most importantly, all of the individuals who've contributed samples to the study. Okay, so I just want to start then by talking about where we are at with genome-wide association studies of inflammatory bowel disease. So far, we've identified over 250 regions of the genome that are significantly associated with IBD, and you would think that would give us a very good insight into the biological mechanisms which are causal of the disease. However, the interpretation of the GWAS loci has been somewhat hindered by the fact that many of these loci, actually the causal variants don't lie in the coding regions of the genome, rather they lie in the non-coding regions of the genome. And so it's difficult often to work out which genes are perturbed, are dysregulated by those non-coding causal variants that we've associated with IBD. And not knowing the effector genes hampers our ability to understand and derive new insights in disease biology, and also go on to identify new drug targets. So we need to come up with a high throughput way of moving from non-coding variant to effector gene, effector cell type, and through to biological understanding. And so we asked the question, can we use single-cell RNA sequencing of disease-relevant tissues in health and disease to help us nominate candidate effector genes and cell types for IBD risk loci? And I'm going to just go through our work to date on that. Oops. So our study has so far recruited 26 individuals who don't have IBD. We've taken biopsies from the terminal ileum. Obviously, they don't have IBD, so those biopsies are uninflamed. We've also recruited 25 Crohn's disease patients, again taken terminal ileum biopsies, but we've taken those biopsies in the inflamed Crohn's disease condition. We've then put those biopsies through a single-cell dissociation protocol that's been developed in the lab by Rebecca McIntyre. And this protocol was developed with the idea of trying to make sure that both the immune cells and the epithelial cells were maintained in our analysis in a happy and healthy condition, so we could look at both the immune compartment and the epithelial compartment, and perhaps even get access to those epithelial cells that lie at the top of the villus, and not just be restricted to those that lie within the crypts. So what have we found in the analysis so far? Well, we've identified 49 different cell types among the 140,000 cells that we've sequenced to date. And here's just an overview of what we found, the main cell populations in the middle of this figure. And then what we've done is broken out those cell populations around the outside and showing you the kind of higher resolution cell types that we're getting across both the immune and enterocyte and secretory populations. And it's those enterocyte and secretory populations that I just want to draw your attention to. So using knowledge of marker genes that exist in the literature from human and mouse studies, we were able to annotate our clusters in terms of the position that the enterocytes lay along the crypt-villus axis. And you can see that we believe that we've got this small population of enterocytes that reside at the top of the villus. And same goes for the secretory cells, being able to place those along the crypt-villus axis and get a population of goblet cells that we believe are from the top of the villus. So we're very pleased about this. It's very difficult to get hold of these cells. How can we try to convince ourselves that we've annotated these cell types correctly? Well, Monica decided to undertake an analysis using SC-Velo, using the RNA velocity method to kind of overlay RNA velocity information on the top of our cell type annotations. So what I'm showing you here are our cell type annotations for those enterocytes and those secretory cells, but with the RNA velocity analysis overlaid on top. And the RNA velocity analysis was done blind to our cell type annotations. And you can see that actually it recapitulates the expected structure of the villus very well. We have up here the stem cell populations coming down into the enterocytes, the middle of the villus, and then ending up with the enterocytes at the top of the villus. And the same goes for the stem cell journey through Panus cells, and then the goblet cells from the middle of the villus towards the goblet cells at the top of the villus. So we're very pleased that we think we've been able to capture the endothelial and secretory cells throughout the villus, and we really look forward to including these cells in our analysis. We think it could be very informative for Crohn's disease biology. Okay. That's an overview of what we've managed to create in terms of an atlas, but of course our aim was to try and understand something about the causal biology of Crohn's disease. And so to do this, we're really keen to try and use that causal anchor that genome-wide association studies and genetics more generally provides biology. When one detects an association between a genetic variant and a disease in this case, you know the direction of effect. Its genetic variant causes disease because that germline genome sequence is laid down at conception and remains stable throughout life. It can't be that disease causes that genetic variant. And so that's a really nice causal anchor into any trait or disease. And it's one of the reasons why I've personally always been so excited and motivated to study genetics. So can we overlay what we've learned in terms of the genetic risk of Crohn's disease with what we've learned from our single cell RNA sequencing? And absolutely we can. So we were keen to ask the question, in which cell types are specifically expressed genes which seem to have their expression somewhat restricted to particular cell types? In which cell types are those specifically expressed genes enriched with Crohn's disease heritability? And you can see the results here on the y-axis. I'm showing you those 49 cell types I talked about earlier, and here I'm showing you the the cell types which were significant in that enrichment analysis. The cell type specificity was estimated using CellX, and then the heritability enrichment analysis was undertaken using LD score regression. And it's, I suppose, reassuring that we find many of the cell types we may expect to play a role in Crohn's disease, Tregs, for example, and monocyte-derived macrophages and monocytes. So now we were keen to ask the question, which genes in the genome are driving the enrichment of heritability seen within these cell types? And so what we can do is undertake a different analysis, this again using CellX to provide us some measures of a gene expression specificity within Tregs, but using MAGMA to partition the heritability that we see from our genome-wide association studies down to gene level resolution. So on the x-axis here, I'm showing you the -log10 p-value from the MAGMA analysis. So you can basically think about as you kind of walk along the x-axis, these genes are capture more heritability of Crohn's disease, and on the y-axis I'm showing you CellX expression specificity. So as you go up the y-axis, these genes are more specifically expressed in Tregs. And what I've done here is just annotated a few of the genes which are really serving as nice positive controls. These are genes that we know are involved in in Crohn's disease risk, largely because we've been able to work out the functional mechanism, or perhaps there's a coding variant underpinning the association. And actually, I just want to point out this LRRC32 example. It's a great paper published in Nature by Nasrallah Immunowski et al. In Goja Trinka and Rahul Rachaduri's lab. And you can see here very much implicating this gene in Crohn's disease, but also then acting via Tregs. So that's a nice set of positive control examples. However, I just want to throw a bit of caution, I suppose, to the wind by showing you this example here. And you can see that there is a gene ADCY7 that in our analysis, in the MAGMA analysis, seems to be capturing a lot of heritability for Crohn's disease. And I think this actually points to perhaps some of the false positive rate of these analyses. And I just want to go into a little bit of detail of why we think this one might be a false positive association. So what I'm showing you here is the association evidence from the GWAS in and around the ADCY7 locus in Crohn's disease and in ulcerative colitis. And what you can see is, if for the time being you only look at the blue points, those are the kind of straight up genome-wide association statistics. You can see that in ADCY7 there's a really strong association in ulcerative colitis, and there's some residual association for Crohn's disease. But in Crohn's disease, what you see with the grey dots, sorry, going back, with the grey dots is there's this very strong association over NOD2. And actually, we've kind of truncated the graph at minus log 10p of 40. Those association statistics go all the way up to 10 to the minus 200 because it's such a strong association. And then that association signal has very strong, broad shoulders, such that even SNPs that lie in and around ADCY7 for Crohn's disease capture some residual association, even though they're in weak linkage disequilibrium. If we condition on those variants that are associated with Crohn's disease in NOD2, you can see via the blue dots here that the association signal in ADCY7 very much disappears. But our analysis in MAGMA doesn't seem to have accounted for the fact that there is this close association between ADCY7 and NOD2, that these two signals are actually somewhat in linkage disequilibrium. So the reason why we've chosen not to kind of put all of the gene names on the points here that we think are perhaps IBD affected genes is because we're very keen to go through now and just make sure that these genes aren't actually being falsely implicated in IBD risk due to linkage disequilibrium in causal variants in other genes being picked up incorrectly in this heritability partitioning analysis in MAGMA. Just to kind of quickly touch on the fact that, yes, we do have cases and controls in our analysis. So one thing we can do is we can perform differential gene expression analysis and then ask the questions about which differentially expressed genes, which cell types are differentially expressed genes enriched with Crohn's disease heritability. And if you do that, you'll see we get, again, this consistent finding of T cells and CD8 T cells and actually CD4 Tregs. So it's a nice consistency of message there, comparing the specifically expressed genes and the differentially expressed genes. Okay, so I'm just going to wrap up there with a few take-home messages. So we've generated a large single-cell RNA-seq atlas of diverse cell types in health and Crohn's disease. I think I've shown that single-cell RNA-seq data can be combined with GWAS results to nominate effector genes and cell types perturbed by these disease-associated variants. But care needs to be taken to fully consider the effects of linkage disequilibrium, and we're still working through analyses and modifications to try and better account for LD in those analyses. It's important to ascertain material from disease tissue to capture disease-relevant cells in the disease-relevant environment. Due to time constraints, I haven't really had time to touch on that, but happy to go into that in the Q&A that follows. And where we're going with this study is we're trying to enroll sufficient numbers of Crohn's disease patients and healthy individuals that we can perform EQTL mapping across and within all the cell types in the gut, so this can aid co-localization of GWAS hits to their effector genes. So I'll thank you for listening, and I'll take questions.
Thank you very much, Carl. That was a really superb talk. So we don't have so many questions, but I'd quite like to just ask one for your EQTL analysis. How many people are you expecting to have to enroll to conduct this type of analysis?
Yeah, so the target sample size is 300 non-IBD patients and 160 IBD patients. So yeah, I think the minimum number of individuals that I would ever advise anyone attempting to do a QTL mapping study on would be 100, and obviously you'll find twice as many QTLs if you have 200. And so you know, we are those are the sample sizes that we have in mind. Those are the target sample sizes. Yeah.
Wonderful, which should provide an amazing resource as well as answering those questions. So thank
the
you so
idea
much.
of having EQTL maps across just those 49 cell types is just so exciting. It'd be amazing.
Yeah, exactly.
And then to be able to compare the genetic effects on, you know, within and across those cell types between health and disease. Yeah, I mean, yeah, hugely exciting.
We very much look forward to that. That's wonderful. Thank you very much.
Thank you.
Okay. So we just move on then to the second talk. This is from Tomohisa Sujino from Keio University in Tokyo, who's going to talk about the characteristics of CD4+CD8aa+Cytotoxic T cells in the gut in Asian IBD patients.
Thank you for having time to talk about my session. So I would like to share my slide. So my talk is a characteristic of CD4+CD8aa+Cytotoxic T cell in the gut in the IBD patient. So IBD patient in Asia is for the Crohn disease and the ulcerative colitis, the patient number is quite increased. However, in Asia, most of the IBD patients do not have the genetic background, and celiac disease is rare in Asia, but Behçet disease is common in Asia. So I want to talk about a little bit about the mouse data. So I focus on the intraepithelial lymphocyte, which is most of the T cells coming from the natural CD4+CD8 cells and also induced CD4+CD8 T cells. So I mainly focus on the induced CD4+CD8 T cells. These cells are working as a tissue repairing and also the protection against infection and food antigen tolerance and maintenance gut homeostasis and microbiota equivalent. So here is some mouse data. OT2 mice, which lacks the rank 3 knockout mice, do not have the CD4+CD8 T cells, giving the over diet induced severe diarrhea in the food allergy model. However, normal OT2 mice, which has a CD4+CD8 cells, which also giving the OT2 cells transfer model, can rescue this diarrhea model. And also last year, the Bilate showed the over-listeria model. So over-listeria model shows OT2 mice, OT2 T cells transferred to the RAGS knockout mice and giving the over diet to induce CD4+CD8 T cells and giving the listeria monocyte, which are composed of the over protein, can protect the invasion of the listeria monocyte to the liver. However, the SFP protein cannot protect to the invasion of the liver. So these data indicated that CD4+CD8 T cells have the protective function to the epithelial component. And we also demonstrated these CD4+CD8 T cells coming from the Treg cells. So here's the intervitreal imaging. The red cells show the Treg cells and X-Treg cells in the mouse model. Some of the red cells are moving like the interepithelial cells, and these cells are coming from actually from the X-Treg cells. So we next focus on how the mouse CD4+CD8 T cells characteristics. So the mouse gut immune cells, we performed the single cell analysis. So in the UMAP shows CD4 cells and CD8 expression, these cluster 1 made the CD4+CD8 T cells. So these characteristic genes, GZMA and ITGAE is a cluster 1, is a featuring gene of the CD4+CD8 T cells. So we next move on the single cell analysis of the human gut immune cells. We collected the colon cancer patient, obtained the normal region, and also UC patient and IBD patient. We obtained the one UC and one Crohn disease. And we collected the tissue and performed the single cell sorting and 5' single cell RNA-seq. So these are the combined data. The several clusters show the 18 fraction, and also clustering in the most of the T cells are coming from CD8 and TCM and CD4 TCM. So here is a map of the each clustering gene. For example, cluster 1 shows S100A1, which is TCM, CD4 TCM, the main gene. And also, for example, cluster 10 shows granzyme B and CD8 alpha. And also cluster 11 shows DATF and IL-2 receptor alpha. These are the Treg fraction genes. So we focus on the CD4, CD8 double positive cells to analyze. However, for example, CD4 T cells in the red dot shows, and CD8 cells shows in the blue, and we merged the CD4 and CD8. However, you can see the CD4, CD8 alpha double positive cells do not categorize in the one cluster. So we changed our mind. So functional classification of the CD4, CD8 alpha cells in human mice. So we collected mouse data to the human data, which cluster is a functional equivalent for the CD4 and CD8 alpha cells. So functional annotation of the human T cells based on the GSFA. So we create a gene set of the each cluster from the mouse, cluster 1 to cluster 11, and then combine to the functional GSFA of the human clusters. For example, human cluster 1 goes to the high relationship to the cluster 5 from the mouse data. And also human cluster 5 has a high correlation to the mouse cluster 4. So here is a map of the cluster 1, mouse cluster 1 to 11 to the human cluster 1 to 18. For example, human cluster 1 is highly correlated to the mouse cluster 5, and human cluster 3 goes to the mouse cluster 5. Then we changed the looking data. So what kind of the mouse cluster goes to the human data? So human mouse cluster 1, CD4, CD8 alpha for T cells, which we are targeted, mostly correlated to the human cluster 1, human cluster 10, and human cluster 2. And also mouse cluster 6, which is Treg cells, are highly correlated to the human cluster 8 and human cluster 11. And then we look through the human cluster 10. Human cluster 10 shows human cluster 10 shows a low CD4 expression. However, the CD8 expression are higher in other clusters. And also, we can see the Crohn disease and healthy control. There's not so much difference between two groups. And also, cluster 10 shows the ITGA are highly expressed, and also HR are highly expressed. And we look through the proportion of the healthy control and Crohn disease in each cluster. Actually, cluster 10 is a lower number of the Crohn disease compared to the healthy control. So these data indicated that Crohn disease is a lower proportion of the cluster 10, same as the mouse model. So we then go back to the mouse model. We look through the human cluster 10, which is expressed the HL signaling. In the mouse model, HL signal is important to induce CD4, CD8 alpha double positive cells. There is a lactobacillus which has a HL ligand inducer, has the amount of the induced a lot of the CD4, CD8 alpha positive cells. However, which lacks the HL ligand, do not induce CD4, CD8 alpha positive cells. Also in the mouse model, HL ligand knockout mice do not induce the CD4, CD8 alpha positive cells. So this means human CD4, CD8 alpha T cells are not categorized in one cluster. However, the human cluster 10 is molecularly and functionally related to the mouse CD4+CD8aa+ positive cells. And the percentage of the cluster 10 is lower in the inflamed colon, UC and CD, compared to the healthy control. So these data indicated that integrating the mouse single data to the human single data uncovers the function of immune cells in the human. Thanks for listening. So this is acknowledgement. So this project is made by the Keoriken HUMED project. I thank to the Jae Singh and Ando Yoshinari, and also the Chan Ho Song and Jonathan Moody. Thanks for the attention.
Thank you very much. That was lovely talk, really kind of investigating those cells further. They are a very small proportion of cells, aren't they, in the intraepithelial site. What do you think they may be doing in human?
I still don't know what kind of they are doing, because it's still unknown in the mouse data. So they are just, we don't know how the exact molecular function. They just induce a lot of the amount of the granzyme A and granzyme B, but we don't know what kind of they are doing.
And if you do receptor analysis, do you have any sort of ideas about what they might be targeting?
I think they are targeting to the epithelial cells. So we don't, last time we didn't focus on the epithelial cells, so we need to merge the epithelial cells, single cell data, and combine the and analyze.
Okay, and I just have one more question. So, which is, do you notice an increase in FOXP3 in some of the subsets at all of these double positives?
In the mouse model, in the human, actually, there's not increased FOXP3 expression in this fraction.
Yeah. Okay, that's great. Well, thank you very much. That was lovely. Great. So I don't think we have any other questions. So I think then, in which case, we'll move on to the third speaker, Keith Wilson from Vanderbilt University. And he's going to be talking about combinatorial single cell strategies for Crohn's disease gut cell atlas. I think you're on mute, Keith. Keith, you're on mute.
Okay, how about now? Yes?
Yes.
Yes.
Okay, sorry about that. Okay, thank you. So this is a multidisciplinary team from Vanderbilt University Medical Center, our medical school, and our Department of Engineering. And for some perspective, I'm a physician scientist, gastroenterologist who's worked on mucosal immunology for many years. But a lot of what I'm going to talk about today is how to think about designing a project like this from the clinical standpoint. So I thought it was important to talk about what was the rationale for embarking on this Crohn's disease gut cell atlas. And this work is being supported by the Helmsley Trust. So just a little bit of background that wasn't mentioned. IBD is increasing in prevalence worldwide, unlike a lot of diseases that are decreasing. And current estimates are 3 million afflicted people in the United States, more than 2 million in Europe, and a very sharp increase in other locations such as China and India as Westernization is occurring. There seems to be a positive correlation. So IBD consists of ulcerative colitis and Crohn's disease, and each has very variable presentations. And unfortunately, the customization of treatment is actually quite limited. Despite a lot of good biologic drugs, there's not very much customization. So a difficult challenge has been to determine at the single cell level the cell types and behaviors and intercellular communications that determine the disease phenotypes. And this project was developed actually in specific response to a request for applications by the Helmsley Charitable Trust, in which they wanted work related to Crohn's disease involving the terminal ileum and ascending colon. So this is very specific. So in order to develop this proposal, we worked out a strategy of having 6 co-PIs. I'm the lead PI, and we broke it up into different cores, kind of like what you might see in a center grant or a program project grant. So the person really spearheading this project has been Laurie Coburn, who's worked with me for many years. She's a physician scientist, gastroenterologist at Vanderbilt. Ken Lau is a basic scientist who's a nationally and internationally known expert about RNA sequencing. He directs all the single cell work. We have a robust biostatistics core directed by Chi Liu, a computer imaging core directed by Bennett Landman from bioengineering, an RNA FISH core directed by Gregor Newert from molecular physiology, and I direct the admin component and the multiplex immunofluorescence. So our overall aims for this project are to construct a human gut cell atlas of the ileum and ascending colon focused on spatial landscape alterations in quiescent and active Crohn's disease versus healthy controls using single cell RNA-seq, multiplex immunofluorescence, and RNA FISH. We want to integrate the data from the prospective collection and analysis of tissues with very robust clinical data, which is an area of our expertise. And we want to disseminate these data to the scientific community. So in order to get started with this and really not have chaos reigning, Bennett Landman's team spent quite a bit of time using computer coding to develop tools for this. So this is just an overview slide from a small publication that is postdoc generated. And at the root center of this is the REDCap, which is a data capture system that was actually originally invented at Vanderbilt. And there's a printer app and barcoding. Oops. So we used a 10-year-old Mac laptop to do this whole project. So we generate barcodes for everything. There's a data dashboard, a data entry system, so that everybody can really keep track of everything. So a big focus has been our accrual. This work started in the late months of 2019. COVID hit in March of 2020. We had no endoscopy for a few months, but Vanderbilt was back online pretty quickly. And our current accrual, excuse me, I thought I was going to sneeze, is about half of our overall goal. So we have control surgical samples. We've enrolled about half, Crohn's disease surgery, and then endoscopy. We have 31 patients out of the 60 desired for control endoscopy, and then 60 out of the 130 that we had planned. And the reason that we have so many is we wanted to have patients at different levels of histologic disease activity. So in terms of what we've accomplished so far with the single cell work, I have 3 different colors here. The top bar is how many everything is done. The next one is how many are encapsulated and waiting to be sequenced. And the last bar is the goal. And you can see that we're doing pretty well considering COVID. If you consider that for Crohn's, we're making good progress. However, a stumbling block is that these are just the Crohn's patients, and the grayish bars are the terminal ileum, and the orangey bars are the ascending colon. And what you can see here is we have a lot of patients that actually had normal histology when looked at microscopically by our research pathologists. So in this project, we don't rely on the ClinPath. We take extra dedicated biopsies that we have our lead pathologist reviews them all, and these are the same blocks that we use for the immunofluorescence and RNA FISH. And quiescent, meaning patients with some chronic changes but without neutrophils, and then mild, moderate, and severe inflammation. So it is very hard to get patients in a tertiary care center who are all on medication. So another problem that we had was optimization of the single cell. And Ken Lau developed some changes where he increased the channel size and he altered the droplet size. And he's the expert on this, so I would refer you to this Dropkick Python package that he uses. If you go to his Ken Lau Lab website, there's a lot of information there. But we had issues with ambient RNA. I don't know if the other teams have. So some of our early samples were excluded. So currently we have 89 sample sequence combining everything. So just to give an idea of what a sample generates in terms of cells, because as I'm sure you know, there's lots of papers in the literature in IBD with a handful of patients and for single cells. So here, for example, we're looking at 41 samples from 22 patients, which is combined data from 2 batches. And you see our distribution by diagnosis, and then you see our distribution by the number of cells. So it's about 75,000 cells from 41 samples. So our bioinformatics group recently generated these data. They've been focusing on the epithelial changes right now, but we're of course going to do more work on the immune cells. And they picked out this, they're excited about this one region, this one cluster here because of some of these innate genes that have implicated inflammation. And so this is cluster 0, and this is the TI, and this is just the enterocytes. And then in these figures, they did some specific gene expression analysis of Crohn's versus normal. And then if you break it out into active versus inactive, you can see a difference. But even with this, we don't have sufficient power to determine the different levels of disease activity, which is why we're continuing our project for quite a while. And then when we looked at the ascending colon, sorry, I don't know why it's doing that. When we looked at the ascending colon, there was also a cluster of some of the similar genes, but now it was over here, cluster 6. And again, with these plots, which are underpowered, but there is some increase in Crohn's for this cluster set of these 3 genes that they focused on for us. So cell type annotation was just discussed in the last couple of talks. I just wanted to emphasize that here, if we look at the salmon color as ascending colon and the teal is TI, there's overlap, but also some pretty clear separation. And when the bioinformaticians worked on the overlay at the cell specific level, they got very interested in these 2 regions for ascending colon and TI, looking at various transit amplifying cells, mature cells, and stem cells, and how they're very different here and here. And you can see the immune markers and tough cells. The UMAP plot is here, but when you look at the heat map, there's a distinction between the TI and ascending colon. And when all the samples were overlaid and we looked at disease activity, you can see this problem that patients with Crohn's but have normal histology clusters closely with the normal control patients, and then the mild, quiescent, and moderate, and we didn't have enough severe to be on here. Another thing that was noted by the bioinformatics group was that when the dendrogram was done of some of these clusters, AC being ascending colon, TI being terminal ileum, that yes, there was distinct separation, but this AC6 and TI0 seemed to cluster closely together, and that's highlighted right here in this square. So multiplex immunofluorescence has been going on at Vanderbilt for a few years in the digital histology shared resource core. And this is just a schematic of this idea that you can use fluorescent markers with validated antibodies and then do multiple cycles, maybe as many as 20 or more, of staining and then bleaching and reacquisition. And these are all whole slides scanned at 20x, and then huge files are generated that go on a server that can be viewed. And so this was some data we generated for the original grant submission, looking at markers and how they may be differed. So for example, a big increase in FOXP3 T helper cells here with the Crohn's patient. So these are some of the markers that we're using right now based on our collaboration with the Digital Histology Lab. And it includes cytoskeletal markers, epithelial markers, stem cells, various immune markers, myofibroblasts, proliferating cells, goblet cells, and enteroendocrine cells. And I think the frustration for me is that it's this issue of the validated antibody. So we can't just use any off-the-shelf antibody. So just a little bit of data. This is a patient with mild activity in the colon in the ascending, and you can see this robust increase in MUC2 that the assay was able to detect. And basically, there's enough resolution that I was able to digitally zoom these way in from the whole slide scan. And then this is a multiplex immunofluorescence for various immune cell markers, helper cells, activated T cells, macrophages, lymphocyte B cells, and monocytes. And you can see this confetti appearing thing because of all the colors. And when I zoom in, you can get, a sense of what this system is capable of. We also have been exploring using validated probes from Dr. Newart's lab. So someone that works in my lab did the CD68 fluorescence in situ hybridization, and that's what it looks like. So what strategies can we use to increase the number of patients with active disease? So we think you have to be very flexible because most patients with active disease will be scheduled on an ASAP basis rather than way in advance. So this puts a strain on our coordinators and a strain on Ken's lab where they have to work on the samples directly and fresh samples. We do review all the charts and look for indicators that suggest they could have active disease. In the endo lab, we use a simple endoscopic scoring system, and if they have active scores, these are the ones that we're really trying to rush to Ken's lab for the single cell encapsulation. And we've also tried to engage our inpatient GI hospital service to let us know when there's a sick Crohn's patient who's going to undergo endoscopy for presumed active disease. So a new adventure is that Laurie, who's our clinical leader, and Bennett, who's our engineering leader, have developed all the plans for an admin core and collaborative data repository for the 11 Helmsley funded gut cell atlas teams. And the goal is to create this admin core and a pre-publication data repository for pre-publication collaboration across all data types. The admin core will provide centralized administrative support to facilitate interactions between the teams, which is very challenging, and to provide support for the data repository. And this data repository will provide technical and policy support for all the GCA grantees to centrally curate, collaborate, and cross-pollinate. We are going to encourage and facilitate sharing of data and biospecimens among the consortium teams through this resource. We're going to maintain the records for data sampling and sharing and documentation of MTAs and IRB approvals and follow up that data sharing has occurred successfully. There'll be an executive committee to evaluate things. And I just want to highlight that we'll have a pre-publication data repository using a cloud-based collaboration. We're going to use open source Novartis cell by gene gateway and server with authentication. There'll be a digital slide archive software on a private cloud for the GCA and offline slide viewing with QPath, collaborative web portal using XNAT, and data contributor PIs will control access to their data sets, and we will support automated processing and quality assurance. So with that, I'd like to acknowledge the single cell RNA-seq core and its members, Bennett's team and his postdoc, Gregor's team, Asim from my lab, Chi Liu and her team, Laurie and our hardworking staff. We couldn't do this without our 6 IBD attending doctors. Laurie and I are both general GI docs who actually work at the VA, so we're completely dependent on these incredible experts who have this huge number of patients, the digital histology research, and this incredible resource, our cooperative human tissue network, directed by a lead pathologist, Kay Washington. That's where we get all the surgical samples from. So I'll stop at that point. Thank you.
Great, thank you very much. That was really a fantastic talk, and what a tour de force in organisation it would appear. So can I was just have a looking, I don't think we have questions as yet, so I'm going to hog them again, I'm afraid, which is firstly to ask, you show some data from epithelia. So are you capturing mesenchyme as well and other cell types? What's your kind of focus on the analysis side of the single cell RNA sequencing data?
So the goal as written was to do anything and everything. And just to be honest with you, like trying to get usable, understandable data from the bioinformatics team is a laborious process. And these data were from a presentation that the postdoc did like a month ago, and they are focusing on the epithelium right now, which is not what we've directed them to do, but that's the data that we have. Ken's group did some initial analyses showing pie charts with major increases in immune cell populations, but I chose not to present that data because it was a different bioinformatics approach. And one of the questions I guess I would throw to the group is some of the teams in the GCA are using CD45 selection strategies to try and increase immune cell populations, but we don't think we can do that and get all the other cells from these tiny endoscopic biopsies. So we had this idea of trying to pilot that on some of the surgical samples, but frankly speaking, most patients with Crohn's now are getting surgery because they have a lot of stricturing disease, not necessarily that they have uncontrolled inflammation. So does anybody have any thoughts about whether the positive selection is necessary, or because we're still waiting for our team to tell us how good their immune cell analyses actually are from the non-selected cells?
So, I mean, my personal experience of this is that basically, you'll get useful information, whatever method you use. And usually that's, it's basically what question you're asking. And, taking a whole biopsy approach, you probably will have difficulty doing like TCR, BCR, repertoire analysis and things like that. But nevertheless, you'll get some very rich data. So, from which you can form a hypothesis, I guess. So I suppose one of the other questions would be, how do you think the single cell data will allow you to actually stratify patients better than histopathology at the end of the day?
So that's a fantastic question, because the question that I was going to ask the first speaker is, when you presented, you just put all the Crohn's or all the IBD in a basket, but do you have, so this is the problem, that if you have, say, I think you talked about 30 or so patients with IBD, if you then try and stratify UC and Crohn's and then normal, quiescent, mild, moderate, and severe, you end up with nothing. And that's the problem that we've learned from previous work in my lab. And that's why we've said 130 samples. Mm. And when we went to the kickoff meeting for the GCA 2 years ago, I was shocked that we had like 5 times as many samples proposed as the other teams. So can you tell me, are you subcategorizing by histology when you analyze your single cell data?
It depends on what we're looking at, but generally not. I mean, there's so many things that you could go after, I think, with it, that was never one of the things that we were looking at. We were initially, because these are studies, I'm talking about myself personally, were very discrete and very different from Carl's, I would say, where, they're looking at genetic risk where numbers may be incredibly important. And so for us, it was more understanding how the immune system was regulating, subcellular structures within the gut. So yeah, I mean, we just wanted a good going colitis. So yes, we ended up taking people who would be at the upper end of the severe spectrum, but not granularly taking a Nancy score or whatever.
So a very firm belief that Laurie and I have guiding this project is that, and that we had to argue about this when we were submitting the proposal, you a priori usually do not really know what the histology is going to show till you take the biopsies and look at it under the microscope. So there's a lot of dissociation, encapsulation, running RNA-seq only to discover that the patient had quiescent histology or was even normal. So that's why I talked about some of the strategies for trying to get more of the patients, but you know, there are non-invasive measurements of disease activity, indices, et cetera, but we're really relying on histo as the gold standard. So, you know, our goal is to do everything. And I think that there's a lot of interest at Vanderbilt in the epithelial cell biology and in the tough cells. Ken has published extensively on tough cells, but I'm more of an immunologist, so I definitely want to know all about the immune cells. And that's why we just need to keep enrolling patients and get as much cell counts as we can for these analyses.
Sounds great. Wonderful. Well, thank you very much. That was really awesome. So I think we're going to wrap up this session now. And just to announce that we've got now a 10-minute break and we'll resume at 11:30 for the orthogonal and complementary approaches session.